Okay, here we go with part 4 of chapter 12. So, in terms of evolution of mitosis, we were talking about the progression of um, very simple prokaryotic binary fission to the eukaryotic steps of mitosis. And we left off with yeast. So, yeast also form a spindle. They also are going to keep their nuclear envelope intact. So while they have the same spindle formation as the more complex eukaryotic animal cells, they are not going to lose their nuclear membrane. And so again, they have to have tunnels in their nucleus for the spindle to pass through and attach to the chromosomes and pull them apart, pull apart the two sets of replicated DNA of a yeast cell. Okay, so let's look quickly at binary fission. So these are the steps, basically, of binary fission. So for example, if we take E. coli, a very common bacteria, cell division is going to start when the DNA starts to replicate at a very specific place on the circular chromosome that's called the origin of replication. And then what's going to happen is there'll be two origins that are produced because that's where replication is going to begin. And as the chromosome continues to replicate, one origin is going to move towards each end of the cell, so moving in opposite directions. And while this is occurring, the cell is elongating. So you can see here there's a stretching out of the cell between here and here. And when replication is complete, the plasma membrane is going to pinch inward to divide the parent E. coli cell into two new daughter cells, each with a complete new genome. It's not new in the sense that it is different, but because it's identical to the parent genome, it's just in two new cells. In most bacteria species, two origins of replication end up at opposite ends of the cell or in some other very specific location. And they think that maybe they're anchored there by proteins. But the mechanism behind the movement of bacterial chromosome is still not really understood. They believe that there are several proteins involved, and some of those have been identified, but there's a lot to learn still about how this occurs because it's also a way to fight pathological bacteria that cause disease. Okay, so now how does the cell cycle in a eukaryote get regulated? So we're going to take a look at this really quickly, but this goes along very nicely with the last chapter on cell communication because how the regulation occurs of the cell cycle is through the use of signaling molecules. The frequency of cell division, as we talked about before, is going to vary with the type of cell. So some cells need to be divided and replicated, copied more often than others, like skin cells, for example. And then there are others that really don't need to, like nerve or muscle cells. Because, for example, muscle cells, our muscles do grow. If you do a lot of exercise, your muscles grow. Well, it's not that you're getting more muscle cells, it's just that the actual cell itself, each individual muscle cell can actually grow. So there's no division really that's occurring with those. But the cell cycle really seems to be driven by a specific chemical signaling system in the cytoplasm. So how do the cells know when to divide? They're going to receive signal proteins that either act as activators or inhibitors. So either it's going to get a signal to move forward into mitosis, or it's going to have a signal that says stay put in that G1, G0 phase. So if we think of the events of a cell cycle as like part of a clock system, we can start to see that there are different checkpoints along the face of the clock, where the cell cycle actually stops until there's a go-ahead signal that's received. And again, the timing and the rate of cell divisions going to vary depending on both the organism and the type of cell, and whether it's for normal growth, if it's for development, or if it's actually for maintenance and repair. So the idea that there are signals that are driving this cell cycle actually came from a hypothesis from experiments where they cultured mammalian cells at different phases of the cell cycle. And then they took two different cells that had two different nuclei at different stages of the cell cycle and fused them together. And so, for example, when they fused an S phase cell, so we're talking about the blue over here, with a G1, this light green phase cell, they found that the G1 cell was induced to start the S phase. So this made them think that and suggest that there's some sort of signaling molecule present during the S phase that stimulates a cell to go from here to here. In this picture here, you can see these like red barriers, and those are really great depictions of the different checkpoints in the cell cycle. So there are probably more, but these are the three major checkpoints. And basically, it's just a place where cell division is going to stop, and there'll either be a go-ahead signal to regulate the mitotic event, or there's going to be a just stay where you're at signal. So these signals are going to be transmitted either within the cell by signal transduction pathways, or they're going to be received from outside of the cell. They really think that that the cell cycle is naturally in the stopped state, and that really what's occurring are go-ahead signals. A lot of these signals are going to come from the actual mechanisms of cellular processes going on, and they're going to tell the cell whether these processes have been completed correctly. So they're literally checking to make sure that integrity of the cell division process is going to continue on without errors. And then these checkpoints also can receive signals from outside the cell. And again, using that signal transduction pathway, the signal is received at either the cell membrane or within a receptor within the cytoplasm that is going to indicate whether the cell should continue with mitosis. For most cells, the G1 checkpoint is called the restriction point. Is going to be the most important one because if the cell receives a go-ahead signal here at G1, it's usually going to continue on and complete the cell cycle through mitosis and actually divide. If the cell does not receive a go-ahead signal at G1, the cell actually exits the cell cycle and switches to a non-dividing state, which is called G0. Most cells in the human body are actually in that G0 phase. And things like the liver cells can be called back to the cell cycle by an external signal, like a growth factor being released during injury, to tell the liver cells to make more of themselves. But again, highly specialized cells like nerve and muscle cells are believed to not divide. Although I have to tell you that I've read recent research suggesting that maybe they're going to be changing their minds about this, that maybe there actually is some division and creation of new nerve and muscle cells. So that may be something revised in the near future. So these signals, what exactly are we talking about? There are rhythmic fluctuations in the activity of a cell cycle. And in terms of the amount of cell cycle control molecules, present, how much is present is going to pace how a cell moves through that cell cycle. These regulatory molecules are mostly going to be made up of proteins, and specifically two types of protein, a protein kinase and a cyclin. Because the kinase is closely related to the cyclin and can actually only be activated by a cyclin, the types of kinases in the cell cycle that we deal with are called cyclin-dependent kinases, or CDKs. One of these cyclin CDK complexes, so when they're bound together, it's a cyclin CDK complex that triggers a cell's passage past the G2 checkpoint into the mitotic phase, is MPF. And so we're going to look at this one a little more closely. It's the maturation promoting factor, or mitosis promoting factor, MPF. So cell cycle controls are going to depend on these things. And so this page actually just gives you a definition of the three players in the signaling molecules for the cell cycle. We have cyclins, which are regulatory proteins, and their levels cycle, therefore cyclins, in the cell. And then the kinases, the CDKs, are cyclin-dependent kinases. And when they're activated, like this one here, here's our CDK. It has a cyclin attached to it in a binding site. This activated CDK then can phosphorylate a cellular protein. Because remember back, a couple units back, we defined a kinase as an enzyme that acts to phosphorylate a protein. So in this case, a CDK is going to phosphorylate a protein that's going to signal the cell to do mitosis. This complex is what triggers the passage through the different stages of the cell cycle past each of the checkpoints. So this picture shows the idea that there are cycling levels of molecules, CDKs and cyclins, in the cytoplasm of a cell. So the kinases themselves, the actual enzyme that drives that cell cycle, is actually present at a constant concentration. So it actually does not fluctuate. But since it needs the attachment of the cyclin protein, it actually is not activated except when cyclin is present. And the levels of cyclin is what fluctuates cyclically. So because of this requirement for binding of the cyclin, again, kinases are called cyclin-dependent kinases. And the cyclin actually is what is controlling whether mitosis occurs or not, because that is what fluctuates. So we'll look at this in terms of MPF. So again, MPF is maturation promoting factor, or some people call it M phase promoting factor, mitosis phase. But we can see that the peak in the activity of MPF is corresponding to the peak in the cyclin concentration. So the purple there is the cyclin concentration. And the cyclin level rises sharply when we enter the S phase and the G2 phase, and then falls abruptly during mitosis. The MPF is what is going to trigger the cell's movement past G2, G2's checkpoint, into the M phase. So see if you can interpret this graph. Make sure you study it and can see how there's a correlation between MPF and cyclin, and MPF becomes active, and cyclin actually is rising sharply during the S phase. So you can see that here. Here's an S phase in blue. We have this sharp increase in cyclin, which is then going to send the cell past G2 into M phase.